I would like to see better, more targeted, efficient gene delivery. I would like to see optimized self-sourcing, and and um, and we're seeing multiple technologies here, and we go back to the core, which is optimize the cell you use and the way you engineer it, so that we have a product, ex vivo, autologous or allogeneic, or in vivo, that it is an optimized product to deliver the value. And those little pieces will make it better, will make it more efficient, will make it less costly, and will give us a lot of value.
Connectivity, I would say. We have a lot of smart devices. I'm really excited about new technologies coming in from manufacturing, from devices, from analytics. But the part which really needs to harness all this power together is connectivity. So we need to think about software. We need to think about how we connect and interchange the information within the systems. And then, we can come to the good place about AI, machine learning, and everything. But if we don't have the data, we don't have the purpose for the data, it's very difficult. So thinking for connectivity by design is very essential, I believe.
I think the technologies need to be not just in the manufacturing space, but we also need to pay closer attention to utilizing advanced analytics. And some examples of that would be instead of doing offline analytics, which takes a long time, we need to incorporate at-line and in-line analytics that they could be non-destructive, non-invasive technologies. They could be something as simple as having sensors in bags, bioreactors. And I think it's also important to use machine learning and AI to make these technologies better and faster so that we're getting results in real time and we are able to manage the manufacturing process better to streamline it, to de risk it in the process development optimization space, but also to achieve the goal of right first time in the manufacturing space.
For us at Nexcel Pharma, one of the things that we have in our bottleneck is providing fill and finish solutions so that we can scale our products. Most of the focus is on actually the numbers of cells that we can generate. But for us, that's not really a problem. Ours is actually the handling in the formulation. So on my wish list is to see new initiatives and new kind of concepts for automated fill and finishing that are suitable for really sensitive cells like mesenchymal stromal cells.
On top of my equipment wish list, there is additional sensor technology. I would like to see online sensors for metabolites, but also maybe for markers to give data-driven information what is going on in my cell culture.
Wow, I have a very long technology wish list, I have to say. Tools a little bit less so. I can't quite spell the word. But from a technology perspective, I think what we are seeing is from gene modified cell therapies, it's improved targeting. How do we think about targeting more efficiently? And how do we think about the persistence that's associated with that? That's one of the areas. I think one of the other big areas is delivery. Having more efficient delivery technologies that's more specific and more sensitive and can get to not only organs, but specific cells within those organs will be very important. I think another area that I think is really, really important is a better understanding of disease and disease pathophysiology on the molecular level. We can target, as we know, genes, and we can target really specific things, but I think our disease understanding at times falls a little bit behind. So that's one of the other massive areas, I think. And then the last one, I think, is an understanding of where we fit into the ecosystem and how do we generate value. For me, that's absolutely critical. So I've got a lot of wishing.
The top of my technical wish list, I don't think there is anything one specific. I think for us personally, for bioproduction at Sekisui, we've invested a lot in analytics and sort of enhancing those capabilities from both early development of scalable, sensitive analytical tools that then transfer seamlessly to QC and product qualification. So anything that brings those two elements to our wheelhouse are welcome.
In terms of technology, I think everybody is thinking of what is the next big gadget. And I don't think automation or machines will fix your issues. I think you have to fix your issues first. You have to look into your process. You have to optimize your processes. And once you optimize your processes, then you can start thinking of the next gadget, automation or something on those lines. So I would like to think that this industry is going to focus much more in continuous improvement, such as lean manufacturing and Six Sigma. It's free. You don't need to invest, right? You just have to utilize your assets when you are in one of those under production valleys. This industry has peaks and valleys. So when you're in production, you are maximizing your assets. But when you are not in production, you have nothing to do. So that is the moment to reallocate your assets, equipment, people, facility, and so on to work on continuous improvement and do more with less.
I think when we're looking at tech and equipment, it helps to look at the process as a whole, the entire manufacturing process that's required to deliver one of these cell and gene therapies. I think to get to a manufacturing process needs input from equipment providers, technology providers, cell and gene therapy developers, CDMOs, and other service providers. It's a real collective thing. And what we're seeing is amazing innovative tech in the space, in the manufacturing side of things. What is really going to have to happen, particularly in the autologous cell therapy space, is some real automation, both in the equipment and kind of process manipulation, and also in the data handling as well. So I think platforms like Cellular Origins, which seek to integrate and automate existing islands of technology and make them automated and link them together, they're going to be critical. Then on the other side of things, all of that data needs to get consolidated, it needs to get processed, it needs to get put into a batch record, which needs to get reviewed and needs to get signed off for a QP by a QP. And when you look at an autologous cell therapy for an indication that potentially has massive patient populations, that becomes a real bottleneck. So looking at systems that enable kind of EQP release are also going to become critical. So systems like the autonomous software platform, which enable that electronic batch record and electronic QP release, are going to have to become critical and will become critical.
Technology equipment wish list would probably be slightly more equitable access to gene editing technologies. At the moment, a lot of IP is held by kind of a small number of parties. And whilst I understand that, there's an imperative to get back your investment in those, it can somewhat limit people's ability to develop new technologies or new therapies. So that would be my kind of number one.
There's a lot of exciting things coming out in the bioreactor field. And something that really excites us at Virica Biotech is we've had a very public relationship and successes with Culture Biosciences, and our ability to affect change within the bioreactor system to move the needle to increase viral titers and quality of virus, especially as it pertains to a bioreactor, has been proven. We actually have a webinar coming up with Culture Biosciences that's going to showcase that quite nicely. So I'm really excited to see that there's a lot more of that coming out on the market. We met with some really insightful startups while here at Advanced Therapies, and there's a lot more coming out in terms of bioreactors that really address the need for specific cell lines. So really pleased to be able to continue to collaborate with these new companies that are bringing, again, more innovation to the space and being able to drive the progress and success, especially as it pertains to viral vector titers.